I am the first science and medicine documentarian to have ever been granted access to this laboratory, which occupies the entire 4th floor, where all the harvesting, sorting, testing, and storing of fetal stem cells occurs daily, to later be delivered to patients on the remaining floors.
The most important part of this floor is the biotechnology laboratory.
Can you tell us what a GMP biotech laboratory, what does GMP mean?
Good manufacturing practice.
According to the U.S. Food and Drug Administration, good manufacturing practice ensures the quality of products by carefully monitoring manufacturers' compliance with its regulations. The regulations make sure the product is safe for use and that it has the ingredients and strength it claims to have, which theoretically qualifies EMCELL to submit to the US FDA for the approval process of fetal stem cells.
And we are the only one in the world which produce the fetal stem cell preparation.
According to the World Health Organization, there is an estimated 40 to 50 million abortions performed annually worldwide, virtually all of which are discarded as biological waste, with a minute fraction of this fetal material being donated locally to EMCELL's laboratory, with the express written consent of each donor. The target time to obtain the most viable fetal material is the end of first trimester.
Our work starts from the moment that they donate it to us. The hospital gives us the fetuses and we transport it here, and then our process starts.
So how long after its arrival? You must act quickly, yes?
We act immediately. It's very important because each minute can influence on the quality of the fetal stem cell preparation. And sometimes we have 1 hour ago it was harvest, and now it's already preparing the fetal stem cell preparations.
Immediately following the abortion procedure, the donated fetal material, along with a blood sample of its donor, arrives at EMCELL's laboratory. Its temperature is checked, and the material is logged into their system. The fetal material is then immediately delivered to their scientists for extraction.
So we put the box inside and need to wait when the air inside of the chamber changed, and then the indicator becomes green.
After the cells are successfully extracted, they are immediately sent to cryopreservation.
The key point of the cryopreservation is to guarantee the percentage of live cells. After our cryopreservation process, we have 98% of live cells in each of our fetal stem cell preparations. So this program is unique because if you look through the other publications for other stem cells, not only fetal stem cells, they have 70 or 80% of live cells, and we have 98. So we're really very proud of it.
After cryopreservation, samples from the new fetal material are tested for harmful bacteria using an FDA-approved BacT-Alert 3D microbial detection system. These tests are done 3 separate times to ensure accuracy. Simultaneously, samples of the same fetal material, along with the donor's blood, are tested in a certified PCR laboratory for any viral infections.
You can see how many tests we have done. And from all of the tests, we have 1 positive for HCV. Скажи HCV. Hepatitis C.
So that fetus, you cannot use it?
No, we cannot use it. And you need to understand that this is not the positive test in the sample of the stem cell preparation. It's the positive test in the donor's blood. And if we even have the positive result in the donor's blood and don't have it in all our checkups in the fetus stem cell preparations, we still don't use it and still discard it.
What is the percentage that you find of fetuses that you cannot use?
10%.
Okay.
Nearly 10% of the fetuses, we cannot use it. There are different reasons for that. There is a microbiology sterility, infection sterility, and also the quality control of the proliferation potency of the stem cells. There is the main three reasons why we can discard the fetal stem cell preparations.
So even if the samples are clean of all viruses and diseases, the viability of the cells are not good?
Yeah, sometimes it happens because of there's different reasons, because not each fetus is the same. They are different, and development of their different, and the donors also are different, and the style of their life are different, and we don't know exactly what influence on the quality of the stem cells proliferation potency. Sometimes we have the sterile sample. The microbiology test was good, the PCR test also was good, but the quality of the proliferation of the stem cells is not enough, so we also discard it.
These tests are also done 3 separate times to ensure accuracy, comprehensively testing for viral contamination. Once the samples have been cleared for safety, they head to the Research and Development Department, where they focus on the quality and viability of the cells, starting with checking their physical characteristics and overall morphology. Using a flow cytometer, they begin to separate the cell types from one another with specialized markers, as well as checking their mitochondrial or energy potential, which is crucial to predict how efficient the cells will behave in the patient after injection. You put one cell and you wanted to test to see if it was going to replicate, and to see how healthy it is, and that one cell created that.
Yes.
This is live, real-time video of a single cardiomyocyte, or fetal heart stem cell, beating in real time that I recorded while there. This is live video of a colony of the same fetal heart cells beating in unison after allowing them to proliferate in vitro, starting from a single fetal heart cell. This is time-lapse footage of the fetal endothelial stem cells, which are responsible for the construction of our blood vessels and building new capillaries to aid in increased blood circulation after they are injected. We are watching, in vitro, the fetal endothelial cells forming their own newly formed vascular system. So you're basically mimicking what it would be like once the cell goes into the human body?
Yeah.
Okay.
We do not cultivate the fetal stem cells.
Right.
We just check up how they replicate in vitro, so then we can be sure that it will be replicating in the organism of our patient.
So it really is about only administering the right amount of cells for each patient, because once they're injected, that happens.
Yeah, very often the patient asks how many stem cells you will inject. Of course, when we are talking about the cultivated stem cells, it's important to know how many stem cells you will be injected, because they already proliferate in vitro and they don't have any potency to proliferate in the body. So, but when we are talking about the fetal stem cells, they can replicate. When other companies use cultivated stem cells, they cannot define their proliferative potential because they already use that potential in vitro. Yeah, and now if we are using the fetal stem cells, not cultivated stem cells, the whole their potential cover up the whole body of the patient, and we don't waste it in vitro cultivation. So that's why the amount of fetal stem cells may be not so high like the cultivated stem cells, but the proliferative potential of them is like 100 times higher when we are talking about like autological.
Very often you hear people say, why do you need new fetuses all the time? Why can't you just cultivate and replicate from one? But you're explaining that it's not nearly as powerful for a therapy, because they're not capable of going into the body and reproducing like the non-cultivated cells.
Yeah.
So what you're doing is basically the most powerful version of fetal stem cell therapy.
Yeah.
Yeah.
And it's not only the most powerful of fetal stem cells therapy, it's the most powerful in whole world, and in whole researches of the stem cells clinical use. Why? Because we have like three types of stem cells: embryonic stem cells, fetal stem cells, and also autologicals or allogenic but cultivated cells. Embryonic is not safe. They can cause the cancer. Everybody knows about that.
Embryonic stem cells are derived from 4 to 5 day old blastocysts after in vitro fertilization. There are no organs or organ-specific stem cells formed at 4 to 5 days of gestation. Embryonic stem cells have a unique ability to develop into any type of cell in the body, which creates a problem: tumor formation. The ability of embryonic stem cells to form non-cancerous tumors, called teratomas, is one of their defining traits. It's also a frightening one, particularly for those who hope to develop therapies from the cells. Fetal stem cells are organ-specific stem cells derived from the tissues of a developing fetus, obtained between the 7th and 11th week of gestation. These cells have already undergone specialization in the germ layers and are tissue-specific.
Fetal stem cells are safe. They are not cultivated, so they don't lose its potential. And the autological or differentiated, for example, mesenchymal stem cells, they don't have any potential when they are injected. So maybe they can influence how in some way, but it cannot be so useful, and it can hold for the years, like we are talking about the fetal stem cells.